you so much for having me out. I'm really excited to come out and share some of my work with you all and tell you a little bit about what I think kind of the future of this sort of thing is going to look like in genomic epidemiology. So I don't think this will be a particularly controversial statement, but let's just say it for completeness. Phylogenetics has been really central to the way we're fighting against SARS-CoV-2, at least with respect to tracking and understanding the movement of the virus around the world. So this is a screen grab from nextstrain.org where you can see kind of this beautiful tracking of geographic structure where the virus emerged in Asia, which is these bluer colors, spreads into Europe. And then of course you can see these streaks of blue where it spread back into Asia. So this really, really fine grained resolution ability to track as it's moving around. And in public health, there's been really two main uses of these kinds of data. On the one hand, you have what we call viral surveillance. So that would basically be identifying a strain or variant of interest and tracking those specifically. So this is a screen grab from outbreak.info where you can see, in the UK, there were many, many different lineages, and then alpha emerged and swept it all away because it was something like 50% more transmissible than the ancestral form. And then, just shortly after alpha seemed to have won, delta come along and just sweeps it out. And now literally everything we ever sequence is delta. So for now it's won. The other main flavor that we see with phylogenetics is what I'm calling genomic contact tracing. So that's sort of a catch-all term that deals with these really fine-grained relationships among samples, where you can zoom in all the way to the tips of my tree and see, for example, that these three are extremely closely related to each other. And the important point about this is it can suggest epidemiological links between samples that you may not otherwise know have a linkage, right? So these three have an identical virus genome. It suggests there's a recent shared transmission history of the virus for these samples. But of course, to be able to do this requires that you have a basically comprehensive phylogenetic tree. The reason for that is you can't possibly know which samples are the most closely related to samples of interest in advance. And so you actually need to have a complete tree so you can see all these really fine-grained relationships if you want to discover these independently of other epidemiological data. Okay. So today I'm going to tell you about two things. So one is our approach for inferring and maintaining a comprehensive phylogenetic tree. To my knowledge, it's the only approach that currently works to do this, but I'm sure this will be a very active area of research for many, many other people going forward. The second part, I'll tell you about how we sort of repurpose parts of that concept to do pandemic scale recombination detection. So going into each of these viral lineages and trying to figure out where we see recombination. And if I have any time at the end, I'll tell you a little about where I think this field is going and what I'm excited to start building next. Okay. So the fundamental problem for phylogenetics and data science during the pandemic is that the scale and the pace of data generation is just insane, right? So this is a screen grab from NCBI Virus, which is a really beautiful portal now for exploring these data, where I'm just showing you the collection date through time. And of course, there's no y-axis, but I'll just tell you this week here, there were more than 100,000 samples submitted to NCBI Virus, or collected and then submitted to NCBI Virus. And so the problem is, of course, there aren't a lot of phylogenetics applications that could even make a tree of those 100,000 samples, let alone the 2-point-something million represented in this plot here. Okay, so the reason this is a problem is that you've got most phylogenetics workflows have been developed sort of for retrospective studies, right? They're very academic questions where we say, okay, here's a set of sequences that we know came from an important outbreak, and let's spend months really dissecting this and figuring out how it works. And so what you would do is you'd gather all your data one time, prune your data, make it really nice, calculate a tree. Which is great, except in a pandemic, by the time I finish making my tree, it's already out of date because a ton of new data has streamed in, right? My multiple sequence alignment has grown. I now have to make a bigger tree. And so, okay, the obvious thing to do is do it again, right? Recalculate the next tree. But you can see where this is going pretty quickly. Obviously, that can't possibly work because by the time that tree is done, there's still more data and it's still out of date immediately as soon as I've made it. And so the problem here is that you can't win this way, right? You will always fall further and further behind because in each iteration, it takes me longer to make a slightly bigger tree. So this isn't going to work. And that was my group's main jumping off point from the kind of existing phylogenetics workflows that we saw was how could we go about writing an application that would be pandemic compatible while we're trying to infer the full phylogeny of all coronavirus sequences. Okay. So this is mostly work by Yatish Taraki, who was a postdoc of mine, and David Hausler at the time. He's now at UCSD running his own lab. And the idea that he and I came to was we could structure our data a little differently. So instead of a tree in a multiple sequence alignment, what we do is we infer a tree, and then we store all of the inferred mutations as what we call a mutation annotated tree. And what that means here is that you can see each branch has a number associated with it. So I store this tree object, but I also store the mutations that I infer to have occurred on branch 1 here, branch 2 here, et cetera. So you can see how this is maybe a slightly better way to do this, because I've actually taken the time to do this whole evolutionary inference, figure out my tree and where all my mutations occur. And now I can store literally my entire history, or my entire inferred history of the virus evolution in this file. So when a new sample comes along, the way we end up, we just add them to this existing tree. So we don't actually go through and do a tree search. What we're doing is a full brute force parsimony search. So we literally check every single branch on the tree to find the one that requires the fewest additional mutations. And you would call it the maximum parsimony placement. So in this case, sample E shares 10U with this branch 6 here. And so it's got one additional mutation that's going to branch off here. And so we put E on our tree. And the really great thing about the way we've structured this is that I can then write this mutation annotated tree file with the sample added back to disk very quickly. And so I can add it when the next samples come in that I want to add more to my tree. I've already got E in there, and I can just keep adding, and it just keeps growing, forever, as far as we know. Okay. One really cool thing about this is it's an extremely efficient data structure, and the reason for that is what we call evolutionary compression. So, for example, a mutation that occurs on branch 4 here would be present in all 3 of these genomes. And in the usual way of storing this, like a multiple sequence alignment or a VCF file, what you'd basically have is a case where you'd have to record that mutation in all 3 of those genomes, A, B, and C. And so, of course, that's not very costly when there's only 3 genomes, but in the coronavirus, there's actually millions of descendant genomes here. And so you want to store that mutation one time, not a million times. And so when we do that, we get this, what we call evolutionary compression, which means that we can write and read this file really quickly, which is a really important thing if you want to add sequences on the fly as soon as they're available to you. As the name would imply, our method is ultra fast. So this is just showing you a benchmark we did about a year ago now, where it took the time to place about 1,000 sequences on a 40,000 sample tree. So these are way out of date, but I'll just say that we initially tried several other methods that were pre-existing for doing sort of phylogenetic placement, and discovered that each of them took more than a day, at which point we said, well, that's not worth anything to me because I could just start from scratch in that time. And that's really how we started making Usher, which took about 43 seconds. So it's several orders of magnitude faster for this problem. And that's why we've been able to keep up with the data as it keeps streaming in. It's also very accurate. So on simulated data and also comparisons to real data, we found that about 97% of the time you get the exact correct placement. That is to say that your user sample that we've added to the tree has the exact same sister samples. And so it's the topologically identical to the correct tree. When it's wrong, it's typically very close. So in that case, it might be something like this user sample branching off from this node instead of this node. But it's in the right basic neighborhood almost every time, and absolutely correct about 97% of the time. So that's very good. I'm going to skip this in the interest of time. Just to show you how this has scaled. So the current big tree is growing all the time, every day. It's currently something in the range of 4.5 million samples, which makes it, to my knowledge, the largest phylogeny ever inferred, which is something I'm kind of proud of and really excited to tell people. And each new addition still takes about ½ second. So we're still actually quite good at getting new samples into the tree. It has taken a little longer, of course, for tons of reasons, but it's holding up. The entire pandemic, so that's sort of everything we know about, is encoded in a file that's about 80 megabytes, which means that it's really great for data sharing. I literally attach this in my emails to send to people anytime I need to, and so it's been great. And I just want to show you, you can actually explore this now. So when we started this, there wasn't really a good tree viewer for the scale we were working on. And when we started on it, we discovered someone else beat us to it. This guy at the Crick, Theo Sanderson, who's just absolutely phenomenal, wrote a program, Taxonium, to display these trees. And you can go there anytime you like. And if you click load the public tree, that'll load up our tree for you. You can explore the lineage structure and all sorts of things. To orient you quickly to how this works, this is delta down here, that's alpha there, and of course all the other smaller lineages up top. So probably the number one thing that we've done with this that's really impacted public health is to make a web portal to run it for people. So the way this works is you can upload your sequences to our web portal. We'll align them, call all the variants, add it to the tree, and then in a handful of seconds, you should get a visualization, which I'll show you in a second. If anyone wants to try this out and wants a little gentler introduction to how this works, this is actually module 3.3 of the CDC Genomic Epidemiology Toolkit. So that's a very, very slow walkthrough I did to show people how to work with this stuff, and I think is generally pretty approachable. This will display at the end is a subtree. So for each of your samples, we'll both figure out how they're related to everything we already know about. So that would be all the black samples in the tree here. And then how your samples are related to each other. So this would be a case where there's 3 user samples that are all closely related to each other and branch off down here from this one sample from Minnesota, USA, Minnesota. So the idea is that you can very, very quickly get this, a handful of seconds, like I said, is how quickly you can get this information and figure out, which samples are related to each other. Maybe you have a nascent outbreak in your community. All these things can be figured out pretty quickly using this kind of approach. Another really cool thing that we decided to start doing is doing cluster tracking for people. So I subtitled this slide, "So we have a tree, now what?" And that's sort of the first question I typically get when I talk to public health officers is, well, what do I do with this tree? And that, I think, is a totally fair question. So one of my students has built this really fun tool to explore these, what we call clusters. And what the basic idea is that it's trying to find a case where a new strain has been introduced into an area and then has grown locally. The idea being that the strains that have come the most recently and grown the largest are the ones you might want to prioritize for further analysis. And so we currently do this at the state level in the United States, and we do that because thanks to the CDC sequencing effort, there's actually fairly consistent sequencing across all of the US. And so you can click the state and see where we think new clusters are coming from. So if you click California, you see it's a lot of Nevada, Oregon, Washington, which hopefully isn't too shocking. There's a geographic component to this, but it's also really nice. And then below this, a table will populate where you can explore each of these clusters in detail, like I'm showing on the right here. And the website for that is right here. You can just search cluster tracker if you like, and it should come up. And of course, if you have feedback, let me know. Okay, so with that, I'll tell you about sort of the second part of this talk, which is how we could detect recombination in these sort of pandemic scale trees. Now that we're making these giant trees, we want to ask the second question is, what happens when trees aren't actually trees, right? So to briefly remind you all how recombination works, the way recombination works is that at least in coronaviruses, and I'm not a virologist, but this is my basic understanding, is that you have replication on a template strand. You have your nascent strand here. At some point, your polymerase falls off, and then it jumps onto a homologous template strand that is genetically distinct from my initial template strand. And so what should come out of this at the end is a new virus genome that's sort of a mosaic of these two, right? It would be sort of pink up until this point and then yellow following this point. And of course, this should be a pretty rare event, or at least detectable recombination should be pretty rare, because it requires that an individual be infected with two genetically distinct strains and they be replicating in the same cell in their body. So it shouldn't be super common, but it definitely does happen. And in fact, we can see it pretty easily if you look at sort of longer term evolution in beta coronaviruses. This is a figure from, I'm sorry, I forget whose paper it's cut off at the bottom here, but basically showing that you get different gene trees or genealogies at different parts of the genome. So for the first maybe third of the genome, you have this gene tree, and then you see it actually shifts here in the middle third of the genome to having Wuhan one up here, but these others that were closely related down here. And that suggests, or is very strong evidence for, a recombination event or a set of recombination events that sort of mixes up the genome over longer time scales. But the question we want to deal with is recombination happening within SARS-CoV-2? And in particular, the reason we care about that is that this could drive the evolution of new variants of concern. And it could also distort phylogenomic and phylogenetic inferences, right? So if you assume you have a tree, but you don't, it can actually mess with the way you're inferring this from the data. So it's something we really need to be aware of and really need to be watching. And just like phylogenetic inference, I think this really needs to be real time so that you can add a sequence to your repository very quickly and determine if it's a recombinant almost immediately. And so that's kind of what we've been working on. So most existing methods to look for recombination in SARS-CoV-2 have suffered from the exact same problem as phylogenetics methods in general, which is that the scale of these data is enormous, and the methods we have don't really work as we would like them to at this scale. And so there's sort of two ways I've seen people deal with this. One is to just consider very specific nodes in the tree. So these Vandesberg et al. Considered the cases where we might look for recombination between sequences in 20b2 and maybe 20a, for example. And what they were basically looking for is cases where you have a mixture of the mutations that occur along these two branches here and here. And of course, you can do that for any set of these, right? 20b2 and 19a, et cetera. So what that means is you're looking for a very specific type of recombination event. And you could actually never see one that occurred between two lineages of 20b2, for example. But it may find some, right? I'm not saying this isn't a valid approach. And so on the other side, there's the other way I've seen people do this is to subsample their data. So in other words, you just take many, many fewer samples, which might work, but of course has the problem that it requires that your recombinant and its ancestors have gotten to a reasonably high frequency in the population such that a random sample from my population has a good chance of catching them, which I think is probably not a great bet because the vast majority of variation in SARS-CoV-2 is quite rare. Okay, so what did we do? Well, basically, I told Yatish that his placement thing was too good. Usher. And so I said, well, let's make it harder. Let's make it an n squared problem. And so the way you can do that is to look for cases where you have long branches on a tree. So for example, sample F here is on a branch with 4 mutations on it. And what you can basically think about is that if there was recombination, this is the sort of signature I would expect because you have a whole bunch of mutations that come onto a genome all at once. And so it's very hard to accommodate that in a tree-like structure because, of course, it's not tree-like. And so what you can do, or what we did, was think of a way to solve this problem where we said, well, look for these longish branches. So I think we used any branch longer than 3 mutations, 3 or more. And then what we'll do is we'll exhaustively subdivide that genome into all single sections and 2 breakpoint sections and place them back on the tree in all possible locations. So totally brute force approach that we just solved with very, very efficient computer science, basically. And so, and when you do that, you discover that there are certain cases like this one where if I put the breakpoint between positions 9 and 10 here in the genome, we find a case where actually no additional mutations are required to accommodate this sample. And so we would say, well, that's a case where recombination is very likely to have caused this because when it's initially placed in the tree, the best possible placement had 4 required mutations. And so we of course, through simulation, determine what our thresholds ought to be and figure out how to deal with this sort of problem, how to determine when we've really found the right places. But that's the basic approach here. Exhaustively subdivide the genome, put it back on the tree. And of course, you can also do that for each of the inferred internal nodes, or even if I don't have a sample from this node, I know what I think that genome sequence was. And so we do it for all the internal nodes as well, because you may only sample descendants of a recombinant and not the actual recombinant node itself. Okay. So just to show you that Ripples does really well, it's very sensitive, it has about a 93% detection rate when we simulate uniformly at random. That is, you grab 2 nodes, you mash them together, you put them back and you see if you can find it. It does incredibly well in 1 breakpoint models. So in black are the subset that we do discover correctly, and red are the subset we miss. You see that we actually do very well. But of course, there's a weak bias such that when my parental genomes are relatively closely related to each other, it's harder for me to get them than if they're very distantly related. I always get them because it's much easier, of course. And the same is true for 2 breakpoint models, but we do a little bit worse for the obvious reason that there's more free parameters and it's a little bit harder to do. Okay, so what do we do? Well, we use this program Ripples to search a 1.6 million sample tree for evidence of recombination. And after extensive QC, so of course, these data are generated in extremely disaggregated ways all over the world in a billion labs with varying levels of expertise. So you got to spend some time cleaning your data at the end of this, but we did extensive QC. And after we did that, we still found 606 what we believe to be unique recombination events. That is, individual nodes on the tree that we think are recombinants. If you take each of those nodes and count the number of descendants, you can get an estimate of how many samples throughout the pandemic have been recombinants or detectable recombinants. In that case, we get a number around 2.7% of all samples. I do want to emphasize this is detectable recombinant ancestry. So presumably the most of the time that recombination happens, it's between genetically very similar viruses. And so of course, it's much harder to find those cases. And so this is almost certainly a huge underestimate of the actual rate of recombination, but it at least gives us an idea how important recombination has been for shaping variation. And finally, if you estimate this parameter that people typically use to sort of summarize the relative importance of recombination versus mutation, which is basically dividing the mutations in a given sample that we think entered through recombination versus the subset for mutation, we get a number that's much less than 1%. So mutation is still the dominant force, of course, but recombination is happening. And of course, as more variation accumulates, we expect to see more detectable recombination events as we go. So some examples of what this looks like. So this is me displaying all of the recombination informative sites across the genome of a recombinant in the middle, and the 2 parents that we're calling acceptors and donors here, where you can see that it has the recombinant for long stretches matches one parent and then matches the other, as we would expect if there was exactly 1 breakpoint somewhere between these 2 mutations here and here. And the same is true for the other cases. This is a nice one because, of course, it was a B117 example. So that means it basically has a ton of unique mutations. It makes it really easy to pick it out. And this is a 2 breakpoint case as well, where we see that the front and back halves of the genome match the acceptor, and then the middle is matching the donor pretty clearly. I do want to emphasize that because there's so little variation when we find these, that the confidence interval for a recombination event is really large, right? It's anywhere between these 2 recombination informative SNPs. So we're not very good at detecting exactly where these occur, but you can still get broad structure across the genome. And when we do that, so now I'm showing you a plot where I've taken each of my predicted breakpoint intervals and shown them as a single gray line, and I'm showing you a density kernel on top of the midpoint of that interval for each of these. You can see this pretty strong structure where the back third of the genome has about a 3 times higher recombination rate than the front 2/3 of the genome. And this is actually really consistent with what people have seen in much longer term evolution in sort of the clade of beta coronaviruses. And so we think this is consistent with the idea that subgenomic RNAs are a big part of what's going on here. And you see template switching onto these because, of course, they're much more abundant in the back part of the genome. And that's at least what my virology friends tell me. As I said, I'm not a virologist. And if anyone else knows the explanation, I would love to hear it. But it seems very likely that something like that is playing a role here. This is also tremendously important because it means that the vast majority of recombination events are going to happen right in the region of the genome that we think may be the most important for the virus adapting to human populations, right? That's the spike gene right there. Kind of the dead center of that is the receptor binding domain, where we see toward the peak of our inferred recombination events. So it's something that we really want to keep looking at and making sure that we're monitoring as virus evolution is ongoing. As I said a little bit earlier, we're using the same sort of design principles for ripples as we use for Usher. The idea is this needs to be real time, right? It can't be that we collect the data and then 2 months later we figure out it might be recombinant. You want to be able to do it now, and so you can. So the way this works is you can add your new sample to your tree with Usher in a handful of seconds, and then very quickly right after that search that one sample and all of its ancestors for potential recombinant ancestry. And this takes just a few minutes, not even on a basic laptop. So we're getting toward where we want to go to with this, where it's completely real time. You get your kind of answers almost as quickly as you produce your data, so that you can keep up with this thing and keep up with the speed of data generation. Okay. In the interest of time, I may skip the back part of this and just go to my acknowledgements real quick and say, okay, obviously I did not do any of this alone. In fact, I think I probably get more credit than I deserve a lot of the time. There's a ton of great collaborators we've had. Some of my favorites are over here. It's Yatish, who wrote the code base, Brian, who wrote quite a lot of the code base and did most of the downstream analysis, and Angie, who built our web portal. And happy to take any questions. Thanks.
All right, Russ, thank you very much. Beautiful talk. So yes, I mean, recombination in the dish we can measure, and it's, as you say, super fast, super easy. Co-infections recombine like crazy. So I agree with you. The limiting thing probably for this happening in the field is the frequency of a person actually getting co-infected by 2 strains. So it's going to happen in super hot zones where there are spreading parental viruses that are having the opportunity to get into some person at the same day, and it happens a lot, obviously.
Yes, yeah, exactly. In fact, I have a figure that basically makes that same point. You need a very specific set of circumstances in order to find these things, and so of course, like I said, it's highly conservative. Like we probably discover, a tiny fraction of actual recombinants using this kind of approach.
So one question I have, so if you let your tree get bigger, built up in your additive way, and then you go back and go take a step back a month or something and rebuild it from scratch, I guess you come out pretty well in the same boat at the end.
Let me see if I have that figure in here as well. The short answer is we don't just add stuff. We've written programs that do tree optimization as well. So what happens is whenever I'm not adding sequences to my tree, I'm actually trying to move branches around on my tree to find more optimal configurations. So, unfortunately, I don't think I have that. Actually, I do have that figure in here. So this would be a comparison of that, where Robinson full distance you can just think of as how wrong I am when I make my tree. And what you're seeing here is a few different approaches for this that people have used. On the bottom is ours, which is we add stuff and we optimize on the fly. And it always does the best when we do these simulations. So I don't think that it is inherently better than maximum likelihood, for the record, but because it's so efficient, I can search a lot more tree space than anyone else can. And that's kind of the secret sauce of why this works as well as it does. But that's a great question. Yeah.